I want to start my presentation with probably what's the most important thing and what I want you to know as you walk out. And I know that's usually put at the end, the results section is at the end. But I would like everyone to know when they walk out of this room today that I am actually talking about zebra mussels and not zebra mussels. So if everyone can just remember that throughout the presentation, it's a surprisingly frequent problem. But anyway, so we're going to talk about zebra mussels, but the first thing that we need to know a little bit about is the biology of zebra mussels. So what are zebra mussels? Well, they're small freshwater European bivalve mollusks, and that basically means that they look like this picture up here. And you can see that they get their common name from the stripes on their shells. It's quite like a zebra, not quite black and white, but dark brown and light brown. They have 3 life stages in their population. The first is a veliger. And a veliger is just a fancy name for a translocating, completely mobile larva population. So you can think of them as zebra mussel young. Once a veliger develops, they grow and they become a juvenile. And that juvenile population is categorized by the development of what's known as byssal threads. And these byssal threads are what allows zebra mussels to attach to substrate. So it allows them to attach to docks, piers, all that sort of things going on. Once a juvenile sexually matures, they become an adult. And then obviously the circle of life completes and adults reproduce and create more veliger population. There's a little bit of confusion regarding the longevity of a zebra mussel population. So they originally are from the Ukrainian area of the Black Sea. And so out there, they can actually have up to about 8 years of longevity lifespan. Around in the United States, they usually typically have only about 4 years. So it's very dependent on where you are in the parameters, in the location geographically. And then of course, we're here to talk about the cannibalistic behavior. So what's causing that cannibalism within the zebra mussel population? And that's actually the filter feeding mechanism. So when zebra mussels are eating and they pull in water, which allows them to pull in nutrients. And that is called filter feeding, and that leads to the cannibalistic behavior because those veliger population is completely translocated. So, what are the issues? Well, the first and the biggest is biofouling, which is the unwanted accumulation of a certain population in an ecosystem, usually an aquatic ecosystem. And the biggest problem associated with that is increased competition for native species. But once again, I talked about those byssal threads. So they have the ability to attach to solid substrate. And once they attach to solid substrate, they can cause physical damage to those artificial structures. They can attach to anything, so wood, concrete, steel. Eventually, with enough time, they'll cause damage to it all. And they can also clog the intake valves of industrial facilities. The city of Monroe, they have a big power plant there, and they actually got caught up in the intake water valve, and it caused the whole area that power plant was providing electricity to actually crash. For a few days, actually. This picture right here is actually a picture of a water level measurer out in Lake Michigan, and you can see it's completely coated in zebra mussels. So, a little bit more. I talked about that filter feeding, and the problems with that filter feeding is that it removes large abundances of phytoplankton, which is decreased, affects the food web, and it also can lead to a toxin accumulation of predatory species. So this has been studied a lot with eagles. Zebra mussels are eaten by fish. Fish are eaten by eagles, and then it actually can lead to decreased reproduction in the eagle population. So what about in the United States? Well, they were introduced into the United States not too far from here, Lake St. Clair, in 1988. And that was done through transatlantic cargo ships. They released their ballast water, and they had veliger population released into Lake St. Clair, and they developed from there. By 1990, they were throughout the entire Great Lakes system. And if I just advance this, you can see how quickly zebra mussels spread throughout the Mississippi watershed, and then also these stars here are by human-induced vectors. So things like recreational boating that happens in the Great Lakes, and then you can take your boat back to California, and then suddenly there's zebra mussel populations. So you can see by 2005 they're pretty much everywhere. But Patrick, this is a math presentation. Let's talk about math, not enough about this biology, right? What this model right here is actually first proposed by an Italian mathematician named Casagrandi, and zebra mussels were originally a problem in Italy, far before they were a problem in the United States. So this is actually his model, and I just want to point out a couple things. The first of which is that you can see that it's actually a stage-structured population. So you have stage 1 is pretty much the juvenile population, and then you have 3 different stages of adults. And you can notice that basically what's happening in these dynamic equations is that you take the population from the previous year and you promote them to the next stage based on some sort of survival factor. So that's what's happening in those bottom three equations. But there's a couple things that I want to point out in this top equation. The first of which is this reproduction factor. And those F's are fecundities, which basically are the average number of eggs released by an adult female per year. For zebra mussels, fecundities can be within the millions. So you have a lot of potential growth in the zebra mussel population based on those fecundities. The second of which is that cannibalistic effect. You see this exponential right here, and that's e to the whatever for people that aren't huge math people. So this exponential is actually adapted from the 1954 Ricker model that used exponentials to model fishery populations. And so you can see that's exactly what's going to control the cannibalistic factor. This capital N of T is the summation of the population, and then that beta factor is a filtration factor. So the higher the beta, the more filtration going on. So what's important about this model? What does it show us? Why do we care about it, right? Well, zebra mussels exhibit lots of different population patterns. So we have three specific behaviors that this model can generate. One's a chaotic pattern, one's a cyclic pattern, and the other is a stable pattern. And it's very important for a zebra mussel model that's generalized to be able to produce all of these, because you'll see all of these happening in nature. And it's not just like in Michigan you see cyclic patterns, and in Ohio you see stable patterns. It's actually, this lake you see chaotic pattern, and then maybe 2 miles down the road you'll see a stable pattern. So it's very, very dependent on not just only the geographic location, but also the parameters that are happening in the specific region. So this model is able to show all three of these patterns. So what did I actually get to do in Texas? Well, I did what I like to call a multidimensional parameter analysis. So we're going to look specifically at the Veliger survival rate, the retention rate of the adult population, and also that filtration rate. So you can see here on the y-axis, I have that retention rate plotted, and on the x-axis, I have the sigma 0, which is the Veliger population survival. And then we're going to move through the beta parameter on that filtration rate, if I just click through here. So you can see what you're actually generating is these striped banded regions of cyclic patterns and then also chaotic patterns. And if I just pull up a specific one, you can kind of see that you have these developed, very, very developed regions of which populations are occurring at certain behaviors, which is something that Casagrandi found. The other thing that Casagrandi proposed was that the beta parameter simply controlled the level at which the population would peak. So a higher beta factor means a lower peak, and a lower beta factor means a higher peak. Well, I decided to look at that as well. And so I generated these. These are going through different beta parameters. So you can kind of see that these are going through different beta patterns, and I did a chaotic one and a cyclic one as well. And so you can see it's kind of right. So these peaks are just changing in height, right? But if you wait for a second, wait for a couple seconds, you'll notice that they start changing in temporal patterns. So the timing on the peaks are changing, which is something that Casagrandi didn't actually look at yet. So I'm going to take a little bit, and that's where my research in Texas actually kept going. So if we go ahead and plot out what I did, which is, here we go. So I decided to look at where those population peaks happened and the timing for those peaks based on the beta parameter. So if you can kind of see, I quite literally just mapped these all out, right, and found out where the top peaks were. And then I decided, let's plot them, right? That's what mathematicians do, we plot things, right? Well, when you're an applied mathematician. So if Casagrandi was correct, you'd expect to see these very, very straight lines all the way across, which you do get to see a bit in the cyclic pattern. But you're also getting these very, very sections right here. And that's when the temporal pattern is changing. So the timing on those top peaks are changing. And that's a result only of the beta factor, because that's the only thing I'm allowed to vary. And you can see it's really, really crazy in the chaotic region. So unfortunately, I'm running out of time, and that's where I have to stop my presentation today. At Texas A&M, we did a lot more research with all this stuff. We looked at an intrinsic growth rate. We also spent some time developing a stochastic model, which I can talk to you about if you want me to talk to you about. But we can do that separately. So I just want to take the time to thank especially Dr. Bogus and Dr. Walton, who were my advisors down at Texas A&M, the National Science Foundation, who is the people that fund REU programs, and actually allowed me to do all this work, and then also Dr. Ross for sponsoring me today. thank you all also for coming. Any questions? Yeah?
Can your model help us kill zebra mussels?
Can my model help us kill zebra mussels? No, my model cannot help us kill zebra mussels. The main importance of what we've done is there's actually little research out there done on what happens to zebra mussels and how the population patterns change. Most of the research is done on, well, how much is this going to cost us, right? So there's a lot of economic papers out there, economic papers, however you want to pronounce it, that talk about, well, okay, by 2020, how much is zebra mussels going to cost us? $5 billion for everyone, and they take into account everything because they surveyed like golf courses, beaches, all that sort of stuff. Like how much is zebra mussels taking? So there's a lot of that type of research done, but not so much on the population patterns. And so unfortunately, if you could find a way to decrease beta parameters and stuff like that, or increase beta parameters and decrease survival rates, then yeah, kill zebra mussels if that was your intention. Yes.
I think you said the mussels came from the Black Sea originally.
Mm-hmm.
What's their natural predator in the Black Sea?
You know what? I don't actually quite know. Most mollusk predators are like otters and things like that. I didn't actually spend much time looking at their origins because I was more concerned about the population patterns and stuff. But yeah, I should know that. Yeah.
Did your research get it? Well, you were talking with the introduction about how lakes a couple miles apart might have very different patterns. So your presentation didn't get into that. I don't know if your research has gotten that far, but is there some explanations for that?
Sure. Well, there's a lot of research that's been done. I haven't done anything myself, but a lot of the literature that I've read talks about how different chemicals can really, really affect not only fecundities, but also that filtration pattern. And so those types of things is what makes it vary so much from region to region. When I first started this research down in Texas, my end goal was to develop a model that actually did spatial stuff. So like where are the zebra mussels within a region, and what happens if we, actually Dr. Ross and I are kind of super branching processes. And so what happens if zebra mussels move and then start producing there, right? But it was just taking, I only had 2 months down there, and so we've gotten into a lot of stuff, but not everything that I wanted to look at. So. Yeah?
What was your data source for what you wound up collecting? And then also, I'm sorry if this is an obvious question, but what did you use to actually wind up crunching your model? Is that Excel?
No, so to actually crunch the model, all the code was written in C, the C programming language, and so we were using the Linux version of C editor. And then all the graphs and stuff were done in a statistical package known as Stata. It produces really pretty graphs, so that's why we used it. But yeah, so the data is actually all theoretical. So I'm not actually taking population data from, I'm not running a regression, so I'm not taking population data from actual physical real-life zebra mussels and then modeling to that. I'm actually just kind of going, well, what happens, what can we think of in terms of how these population patterns go? And then it's kind of like backwards fitting, if that makes sense. So not taking real stuff and going to theoretical, but taking theoretical and going to real. Cool? Awesome. Any other questions? Yes?
How would the filtration rate and the fecundity be related? Would that depend on the local environment much, or are they always linked?
The filtration rate, they're not always necessarily linked. It's like I said, it's based a lot on the chemicals that happen in, and I don't know much about it, unfortunately, because I've only read papers on it. And what I understood from it is what it is that you have this filtration rate and these fecundities. The fecundities, I think, are a little bit more sensitive based on like location and stuff like that, because you'll see that fecundities in the United States are actually lower than fecundities in Europe, and that sort of thing. So, did I answer your question? Maybe skirt around it. All right. Anything else?
We have to let you off at this point.
Sure. Thank you so much.